Hey, hi everyone. Get a timer going. I like to talk. You'll soon see. All right. So my name is David, and you all know I'm interviewing for a job. So let me kind of contextualize the work here. So this is going to be a conversation and a presentation about vaccines, and I'm going to try to hit on several of the areas that I work in this. So this isn't going to be a talk that goes into great depth into any one particular topic, but tries to give you a bit of a flavor of the types of projects I'm interested in, the things that I work on, and kind of wrapping that all together under this title of statistical learning for understanding whether and how vaccines work. So I'll start by saying there are a few technical slides in here. I put asterisks on the more technical slides, and I'll spend a little time going through those, time permitting, but those might be things where I move rather quickly through them in the name of talking to you guys about more interesting science. But if you do want to spend a little more time on the technical details, I'm more than happy to slow down and go through that in greater depth. And so with that said, please ask questions along the way. In addition, I'll try to save a little bit of time at the end so we can have a dedicated time for questions. And if you have anything that you think of tonight and you simply must have answered, my contact information is shown here. So one of the fun things about working in vaccine statistics over the last couple of years is that I no longer have to draw silly pictures in PowerPoint to tell about my research, because great people like those at the Washington Post draw much better pictures than me, and I can then steal them and use them in my presentation. So I'm starting here just giving a high-level overview of the way that vaccines work. And this is going to transition into talking about sort of how do we do statistics to figure this all out. So this is a diagram showing a protein subunit vaccine. So this is just one class of vaccine. The Novavax vaccine is built this way, for example. Most seasonal flu vaccines are built this way. And the way this works is we grow these proteins. In this case, this is for SARS-CoV-2. We grow these proteins in a lab. I think for the COVID vaccines are grown in grasshopper cells. So it's some kind of science fiction where we grow these proteins, and that's what's really constituting the vaccine. So when you get an injection of these proteins, it triggers an immune response, right? And that immune response is what ultimately provides you protection from getting infected with the pathogen and ultimately getting disease and the very bad outcomes. But let's look a little bit closer at that sort of immune cascade that happens once you receive a vaccination. So when you get jabbed in the arm, we're inserting these, in this case, little bits of viral particles into your bloodstream, and little cells called antigen-presenting cells pick up those little particles and basically eat them. They digest them, and they basically spit out a little bit of the viral particle and send that on to a helper T cell. And the helper T cell is really the guy that makes then everything happen. And the helper T cell goes off and talks to other T cells that learn to be cytotoxic, so they go off and kill infected cells or cells infected with this pathogen in the future. And they also produce B cells. And B cells are particularly important, we think, for COVID vaccines and many vaccines, because B cells are what produce antibodies. And so antibodies are what's shown in these little Y-shaped figures. They're these little proteins that basically float around in your blood and are made to bind to the pathogen that they're designed to protect you against. So the next time that you get exposed to it, you have these little floating Y guys in your blood. They stick to the virus, let's say, and thereby prevent that virus from infecting your cells. Focusing in on antibodies a little bit more. So antibodies are very sort of particular about what they're allowed to bind to. So what I'm showing here, this is a bit of a surface protein. So you may have heard of the spike protein for SARS-CoV-2. This is basically the equivalent for HIV. So this is an envelope protein of HIV, and it's showing a particular subunit of that. That's here on the left of the figure. And then on the right is a particular antibody. And these antibodies are highly specialized to bind to a particular part of that protein. And so you can think of it as basically your body making sort of the perfect puzzle piece to fit onto that viral protein. So you can imagine kind of taking that, what's labeled VRC01, that's an antibody. You can imagine taking that and kind of sticking it to the protein in that region. And that's what antibodies are designed to do. They stick to things, and by sticking to things, they prevent that protein from, say, grabbing onto your cells and infecting them. So here's an example of where antibodies can start to go wrong. So does anybody recognize this picture? Name that trimer. This is Omicron. So this is the Omicron spike protein. And I'm trying to highlight here what causes vaccines efficacy to diminish. And as we've all learned, the original vaccines that we've developed against COVID for like Wuhan-like strains are not quite holding up as well as we might like them to against Omicron. And why is that? Because Omicron has mutated its spike proteins to, in particular, avoid antibodies sticking to them. So what's shown here in panel B, all the red little bits there are areas that Omicron has mutated relative to the original Wuhan strain. And so as it mutates, that actually changes all of the physiochemical properties of the protein. So the way that the protein folds is different, its shape is different. Whether certain regions are sticky or not, whether the antibodies can stick to them, that's all changed. And so what's shown in panel D there is just highlighting that what's in red is another antibody, and it's showing that it can no longer bind in the way that it previously could because of the mutations that have happened in Omicron. And so we have sort of related statistical frameworks for learning about these phenomena. And these are what I'm going to be talking about in this talk, largely two of related areas of research that I work in, vaccine correlates of protection and vaccine sieve analysis. And this slide is making the point that really these are just two different views of the same biological phenomenon. So what are these areas? So correlates of protection are vaccine-induced immune responses like an antibody response that are causally related to an individual's risk of infection or clinical disease. And sieve analysis, on the other side of the coin, is the study of whether and how vaccine efficacy depends on the genetic or phenotypic characteristics of a pathogen. And so these are, again, just two ways of looking at the same problem, because of course, we know that as things like Omicron emerge and we have mutations, that we expect the vaccine efficacy maybe will be lower. But why is it lower? Because those immune responses are no longer working in the way that we want. So we would do a sieve analysis if we wanted to understand the amount of that efficacy drop-off, and correlates of protection would tell us maybe what the biological cause of that drop-off in efficacy might be. In particular, perhaps your antibodies are no longer binding to the spike protein as well. So depending on the scientific question that you're interested in, you might take a different tack to coming up with a policy-relevant scientific question. And so what are the benefits for sort of understanding both sides of this coin is that, we can learn a lot about how vaccines work and translate that into better vaccine development and better public health policy. So for example, if we understand which immune responses are causally related to protection, well, that tells our colleagues who actually make these vaccines, it gives them a much sort of faster process for developing those vaccines, right? So if you know in early phase clinical trials what immune responses you're targeting, that makes it a lot easier to come up with go and no-go criteria for advancing vaccines to phase 2 and ultimately phase 3 trials. These can also accelerate the approval process, the regulatory approval process for vaccines. So if you understand which immune responses and what level of those immune responses are predictive of vaccine efficacy, then you can potentially start to use those immune responses as a surrogate endpoint in clinical trials that evaluate these vaccines. And the FDA and EMA and other regulatory bodies have sort of accelerated pathways for approval based on these surrogate endpoints. For example, this is how seasonal flu vaccines are approved every year. We don't have time during flu season to run a new clinical trial every single year to decide what flu vaccine we should give. Instead, there's an accelerated approval pathway whereby companies can show that their vaccine produces a certain amount of the right immune response and is safe and gain licensure and approval in that way. And then, of course, sort of health policy, if we understand, which mutations along let's say the spike protein are likely to lead to decreased vaccine efficacy, well, that can help inform health policy as well, right? As soon as Omicron came onto the scene, we sort of already had an idea of which areas of the spike protein were sort of important for antibody binding. And as soon as we saw this, in particular, lots of these mutations down here were in particular regions of the spike protein called glycans. That's like a type of sugar. Sugars are sticky. That's what antibodies like to stick to. And as soon as we saw Omicron emerge and had sequences for it, we knew that this was likely going to be something that could cause problems for vaccines. So by having that understanding, we can inform all sorts of health policies and control policies that we need to try to stem the pandemic. So I'm going to talk about these two sides of the same coin and highlight a few of the projects that I'm doing in each. Again, this is just sort of a flavor of these types. And in periods, I'll try to go into a little bit more depth, time permitting. So we'll start with vaccine sieve analysis. So remember, this is studying basically how pathogens' genetic makeup or phenotypic characteristics impact whether vaccines work at preventing infection and disease. So we're to the the PowerPoint picture side. This is not a Washington Post picture, unfortunately. So where does the nomenclature derive from? Sieve analysis comes from imagining sort of the vaccine-induced immune responses as acting like a kitchen sieve or a strainer, right? And the idea is in the wild, you have all sorts of circulating pathogens. So here I'm showing these by yellow, blue, and green pathogens. In reality, maybe these are something like, your alpha, betas, omicrons, and deltas of COVID, or maybe this is characterized by particular mutations in a particular protein, what have you. So there's some genetic variation that's out there in the population, and the idea is that this vaccine is throwing up an immune barrier to infection by these different pathogens. And if a Sieve effect is present, that means that basically the vaccine is selectively straining out some of these pathogens from breaking through. So for example, maybe we make a vaccine using a Wuhan-like strain, that's what we did, and we find that that vaccine has incredible efficacy at preventing infection and disease caused by Wuhan-like SARS-CoV-2. But then we find as the virus mutates that there's still breakthrough infections, perhaps by different strains. So that tells us we can do things like what's happening now. We're making multivalent booster vaccines. So if we can sort of increase the genetic diversity in the vaccine insert, the antigens that we put in the vaccine in the first place, we may be able to boost the overall efficacy because now we're sieving out more types of the pathogen. It also is important for understanding where and when to deploy vaccines. So this is a bit less relevant for COVID, right? Because we want to deploy vaccines everywhere all the time. But for something like malaria, which I'll talk about, there's real questions about should we spend money on vaccine deployment or should we spend it on bed nets, for example. And if you understand whether these Sieve effects are present and you understand the geographic distribution of the malaria parasite, then maybe you can be smart about the way you spend your resources and allocate that vaccine to where it's likely to have the highest impact. Or similarly, here you can think of this story as sort of saying, here on the right is back in 2020 when we were in Wuhan in D614G world, where we hadn't diverged that far from the original SARS-CoV-2 strain, and now we're in this sort of world here, which is basically dominated by variants of Omicron, and therefore a world where the original vaccine's efficacy has been diminished. So that gives us, again, policy relevant information for making decisions. So that motivates the idea of Sieve analysis, right? We're looking for these type of effects by trying to develop a statistical framework whereby we can analyze data that roughly look like this, where we have some vaccinated individuals and we see what types of infections are breaking through. And we analyze those data in such a way to understand what are these problematic strains or genetic features of the pathogens that cause this diminished efficacy. So we can set this up in a couple ways. So the first thing you might say is, well, just based on your picture, one thing we could do here is just look at the right side of the screen, right? Look at the breakthrough infections and compare breakthrough infections distribution amongst vaccinated individuals and amongst placebo individuals. What you'd expect is individuals who receive a placebo would have a lot more of the yellow pathogen breaking through, right? And whereas individuals who are vaccinated, those yellow pathogens have been sieved out, so we'd see more blue and green pathogens. So that's certainly one way you could approach this problem, but statistically maybe less than ideal, because sort of here, if you're conditioning on people who were infected, you've sort of broken potentially randomization that was was by design in your study. So if you're studying this in the context of a randomized trial, we kind of like to respect that randomization in the way that we analyze these data, rather than conditioning on this post-randomization event, which complicates our lives as it relates to making causal inference. So causal inference is sort of, a foundational cornerstone of this area is how can we set up sort of easier causal estimation problems or causal estimation problems under weaker assumptions that allow us to learn about these Sieve effects. And so that's what I'm describing here is the way that we do this is by studying estimands that look like the following. So we try to mirror here, when we do these Sieve analyses, we try to mirror the primary efficacy analysis of a clinical trial. And so this is really happening as sort of a secondary analysis of a clinical trial. We work with like Moderna, for example, and they need to do their primary analysis to submit to FDA and get their vaccine approved. And then they give us all of the sequence data, and we try to set up an analysis that addresses these Sieve questions. And so in a primary analysis, you might characterize vaccine efficacy in the way that's shown up top here. So vaccine efficacy, as we've all learned this year, is presented in this sort of strange way. It's 1 minus basically a relative risk. And here I'm presenting that risk in terms of a cumulative incidence. So if we imagine T here is time, let's say number of days since your second dose of a COVID vaccine until the day that you come down with symptomatic COVID-19, for example. We're comparing the cumulative incidence for individuals who are on the vaccine versus on the placebo. And we have 1 minus a relative risk, making a VE of 1 sort of a perfect vaccine, a VE of 0 a worthless vaccine, and a negative VE a harmful vaccine. So here I've written this estimand in terms of counterfactuals. So we have sort of T of 1 is imagining what would your time to COVID have been had you been vaccinated. T of 0 is the the contrast to that. What if you had gotten a placebo, for example? And so the way we set up the Sieve problem is we look at that primary estimand and then we sort of make it respect that there may be different efficacy across different strains. So to do that, we use a competing risks framework where we basically say after you get vaccinated, you're at risk for infection or disease caused by a number of different types of pathogens. So I'm using the variable J here to denote what type of pathogen it is that's infecting you. And again, coaching this in the language of counterfactuals. So what we're comparing here is basically the cumulative incidence of a particular type of virus, let's say, infecting you on vaccine versus placebo, and thereby coming up with this sort of strain-specific vaccine efficacy estimate. And this is what we're going to be trying to target as this causal estimate at the bottom. So if we want to sort of test for the presence of Sieve effects, we can set that up in a number of ways. And it's sort of one of the big questions we have when we do this type of analysis is, what are the strains that we're considering? And I think, we're all very used to, talking about now like Delta versus Omicron. And certainly we can set up sort of Sieve analyses that look at that. So Delta, is the VE against Delta the same as VE against Omicron? Lots of papers written answering that question over the past few years. But oftentimes we want to get a little bit more granular, right? We want to look at specific subregions of proteins, for example, maybe this mutation on this glycan, right? Is this mutation really problematic or not for us or not? So we might look at sort of, saying strain 1 is a pathogen that has, or a virus that has this particular glycan at this region, and strain 2 is one that has any other sort of amino acid sequence at that region. So this is a sort of a huge topic for debate, is how do you set up the structure of these strains and what you're calling different strains. So we can also not just look at particular regions or particular strains versus others, we can also look at more like count-based kind of strains. So how far has a given strain diverged from the Wuhan strain, let's say? So we look across that whole spike protein and we count the number of red dots basically that we see in it. And then we might be asking a question like, how does VE vary as the strain diverges from the original strain? Is it sort of like one mutation, any mutation means there's little efficacy, or is it a more sort of granular fall off? So we have sort of similar null hypotheses that we could test for order categorical marks like that. And we call that sort of counting of strains Hamming distance.
That's a good question.
Yeah, please.
Go back to your competing risk little formula 2 slides back. So when you have the bottom formula, so you have J1 equals little j. So let's say it's someone who gets disease symptoms at time tau, you can tell whether it's due to Omicron or Delta. Is that really the case? You can tell, but what?
Hopefully, right? Maybe not in the US, but in clinical trials, we often collect these data. So when individuals are infected or they have symptomatic disease, we'll take a blood draw and we can perform PCR to sequence.
It's always just one, it's always a unique set of, it's either Omicron or Delta. It can't be, well, you got infected with both.
No, I mean, we get a whole sequence, right? We have a whole amino acid sequence for the spike protein, basically. And then you can collapse that into, Omicron versus Delta, or you can look at particular amino acid residues and say, is this a Wuhan amino acid or is it a different amino acid, let's say, right? And this is a real problem, I would say, with the way we're capturing data now, at least in the US, is we no longer get this J. And so we can kind of guess based on time period, but there's not a lot of good data capture systems to get this information. But we often do try to bake it into clinical trial protocols, when the companies let us, essentially. Okay, so the name of the game here for Sieve analysis is we need to estimate the strain-specific cumulative vaccine efficacy. What are the ingredients of that? It's estimating a cumulative incidence. So if we can estimate this cumulative incidence under vaccine and under placebo, for each type of strain that we're interested in, then we can do all the sorts of testing of the Sieve null hypotheses that we like to do. So this is a very, of course, classical problem in survival analysis, right? It's, like going back to the Allen-Johansen estimator, which is kind of the Kaplan-Meier extension to competing risks. But here, just coaching it in the framework of causal inference and asking for a little bit more from the estimator than maybe something like Allen-Johansen has to offer. So in particular, we're interested in developing estimates that can be used both in randomized studies, where we don't have to worry about confounding, and observational studies where we do. So we need to worry about the types of individuals who get vaccinated versus not, because certainly over the last years reminded us that those are maybe very different types of individuals. The second thing is we'd like estimators that are robust to informative censoring. And this is like a real issue, I think, in many studies of vaccines. So for example, in HIV vaccine studies, we're often recruiting, high-risk individuals, sex workers, intravenous drug users, and those are the same types of individuals who are simultaneously at higher risk for HIV infection and also higher risk for dropping out of the study. They're harder individuals to follow up over, the series course of the study. Also in COVID-19 clinical trials, if you'll remember, there was a huge amount of censoring, and it definitely was informative, right? In 2020, there were about 5 to 7 major phase 3 trials in the field at the point that Pfizer and then ultimately Moderna were granted EUAs. And people dropped out of those trials almost immediately, of course, to get that vaccination. But because of the way the vaccines were rolled out, it wasn't sort of uniform or random in the way that they dropped out of the trial, right? Older adults and individuals who are at higher risk for COVID dropped out first. So at a certain point, if we want to use the data from randomized trials to learn about these type of estimates, if we want to extend the time frame long enough, then we definitely need to worry about adjusting for informative censoring. The second thing here is in the context of a pandemic, when we'll get to the point where we're talking about what quantities we need to model in order to estimate and identify these things. And we'll find in the context of a pandemic, it's a pretty complex process that you're asked to model. Different strains emerge at different places over different time frames. And that can be a pretty difficult thing to try to capture with a fairly restrictive statistical model. So basically, we're asking for estimators that can be as flexible as they need to be to accommodate what we know about the sort of transmission dynamics of the disease that we're studying. So a bit of notation here. So I trained at Berkeley, so I use W for baseline covariance. So this is stuff we collected trial enrollment. I use A to denote vaccine status, and then it's sort of very standard competing risk survival analysis notation. So T tilde will be a right censored time to endpoint. So that'll equal your actual time until, asymptomatic COVID, let's say. If we observe that, otherwise it'll equal, the day that we last saw you under study. And then we'll have the delta T that indicates whether, that T tilde corresponds to an event time or censoring time. And then J we'll assume is a categorical strain. It's possible to extend this to continuous sort of strains, but we'll stick to categorical for this talk. And also another simplifying assumption here is I'm going to assume that because this is realistic, that the follow-up time here is discrete. So like number of days until you get COVID, which, all of this, of course, works with product integrals and all the elegance of survival analysis. But for simplicity, I'll work in a discrete time frame. So what else is important here? So yes, N is the sample size. I will denote this whole vector of observed data by O for observed data and say that that's sampled from some distribution P. Well, lots of math on this slide, so let's signpost it. Okay, so the way that we typically go about identifying and ultimately estimating cumulative incidence, a very common approach considers cause-specific hazards. So cause-specific hazards say basically if you've remained uninfected or with no disease up to the previous day, what's your probability of getting disease on the very next day and getting disease caused by a specific type of pathogen? Okay, so it's sort of the standard hazard, but asking for a little bit more, that not just that you were infected, but that it was with a specific type of pathogen. So we'll consider the sort of conditional cause-specific hazard function here, so conditioning on vaccine status and baseline covariance. And under certain assumptions, which I'll sort of wave my hands over the next few slides, we can identify this using a rather straightforward identification formula, where on the inside you have sort of the very familiar, if you work in this area, form of cumulative incidence, the one that like Nelson Ayland and Ayland Johansson estimators are based on. And then on the outside, what's happening is we're just integrating this conditional cumulative incidence over the distribution of baseline covariance. And here I'm just noting this S I've introduced is just a function of those hazard functions. It's the survival function. Okay, so these are the assumptions. I don't want to spend a lot of time on them because they're kind of standard causal assumptions. So we have things like causativity. Everyone has to have a chance of getting the vaccine and placebo. Everyone has to have a chance of remaining uncensored. There's sort of fundamental causal assumptions like no interference. We can debate whether that's appropriate in this context, but I maybe don't want to go there right now. And then we have randomization assumptions, of course, too, which is basically saying, have we adequately measured enough covariates to control for confounding? But I think I'll wave my hands over those in the interest of getting to things that are a bit more interesting and unique to the research I do. So pretty standard set of causal assumptions under which we can identify this estimate. So in a paper now published several years ago, let me see if I can move this out of the way. A paper several years ago, we proposed estimates that sort of try to check the boxes that I asked for. based on the framework of targeted minimum loss-based estimation, or TMLE. This is a real cornerstone of their types of research that I do. And these estimators have some nice properties. So you can show that they're non-parametric efficient amongst the class of estimators, regular asymptotically linear estimators. What I kind of like about these is they're particularly agnostic to the types or the types of models that you're using to estimate things like a cause-specific hazard distribution. So if you like something like a cause-specific Cox model, you can plug that in as a working model in this formulation. If you like something even simpler than that accelerated failure time model, you can plug that in. If you like machine learning, we can also plug that in. So our paper, we describe approaches for using ensemble machine learning, which are really nice for settings where we don't understand a lot about the relationship between covariates and risk of disease, and are sort of more agnostic to which model might perform best. And so we try to learn through cross-validation which model is performing the best. But in any case, that's again, not really the focus here. I like these because they're particularly agnostic. They say, if you like this model, you can use that. If you like fancy machine learning, you can use that, and that's all okay. They end up having a double robust property, as many sort of causal estimators do. And they have a nice asymptotic normal distribution, and we can actually get the variance, asymptotic variance in closed form, so that we can do things like get closed form confidence intervals, for example, a closed form hypothesis test. So this is all wrapped up in an R package that's available on CRAN as well. So I'm going to give you just a little flavor here of how this works. We first estimate, so TMLE unfolds in a few steps. So first, we estimate certain key nuisance quantities. And so we've already sort of seen these on the previous slide. A couple of these, the cause-specific hazards, we're going to need an estimate of that. So fit your favorite model of cause-specific hazards. We're going to need an estimate of the covariate distribution. And that's what we need to sort of evaluate the estimand that we're interested in, right? We use the hazards to come up with a covariate conditional cumulative incidence, and then we average that over the distribution of covariates. But also in the form of efficiency theory and the types of procedures that we need to get closed form inference for this, we also need to concern ourselves with a couple other nuisance quantities. The first is a propensity score. So we need to understand the probability of vaccination as a function of covariates, as well as the censoring distribution. So again, fit your favorite models for these, estimate them in any way that you can scientifically justify. And then the TMLE step is happening in step 2. So we're applying sort of a de-biasing algorithm to these cause-specific hazard estimates that's going to remove sort of residual bias that might be incurred from model fitting. And I'll briefly flash a couple slides that will show a little bit about the underpinnings of this. And at the end of the day, once we're done, we have this de-biased version of the cause-specific hazard functions. We have our estimate of the covariate distribution. We can just generate a simple plug-in estimate based on those.
Right. So J has disappeared from that.
Yes.
Slide. Is that sort of all conditional on J, or is J on the left-hand side?
Yeah, so J is one of these different ones, right? Because we need to estimate the whole survival distribution here. So to estimate the survival distribution, we need the cause-specific hazards for each type of strain.
So when the covariate distribution is conditional on J too, is it?
No, that's the marginal covariate distribution. Yeah. Okay. All right, so I think I'll go pretty quickly through the estimation theory. This is just showing you a little bit about how we study these, and then getting a little bit into the general theory that I use in a lot of the problems that we have. So this is basically just showing everything that I said in words on the last slide. We need to fit estimates of what we call nuisance quantities. These are sort of the intermediate building blocks that we need to get our final answer and final inference on the quantity that we're interested in. And the way that we study the theory of this is by using non-parametric and semi-parametric efficiency theory. As part of that, we are interested in characterizing the so-called efficient influence function of the parameter that we're interested in studying in the model that we've chosen. And once we have that efficient influence function, we can sort of provide a linear approximation. It's basically a very fancy Taylor expansion. So we provide a very fancy Taylor expansion that provides an approximation to this parameter. And that approximation, this linear approximation, is sort of the foundational building block for how we study the asymptotic behavior of this. So this is too much for this talk, but I'll just mention a couple things that come into play here. So in order to study the behavior, we need this linear approximation, and then we need a couple sort of additional pieces from the literature to help us characterize the asymptotic behavior. So these are things like empirical process theory coming out of very smart people like John Wellner and Odd van der Vaart that teaches us when we can ignore certain terms in this linear approximation. And we also need certain convergence rates. So yes, you're allowed to fit any nuisance model you like, but there are, of course, regularity conditions on which of those will lead to nice asymptotic theory. But we can characterize them in the form of these two assumptions. They involve the sort of like L2P convergence rate of the nuisance parameters, as well as sort of characterizing the whether these satisfy the certain key empirical process condition called the Stansker condition. And then ultimately what the study of these estimators boils down to is the study of two terms. If we have these sort of assumptions that hold, we have basically the estimation error of the parameter that we're interested in estimating behaves like the sum of two terms and then a bunch of stuff that we can ignore because it disappears asymptotically. And these two terms are basically something that involves the influence function that's here. And this is a very well-behaved term. It's just a sample mean. And we all know how to study sample mean because we all know the central limit theorem and we all know the weak law of large numbers. So we're very happy with that green term. It's this red term that ends up being particularly problematic and why we need this sort of iterative debiasing algorithm. So this is a bit of a nasty term here because it's a mean, right? But it's a mean of this sort of random function. So you have your sort of data entering into this mean in two places as a part of these estimated nuisance parameters, as well as when you're evaluating the value of this. And that can cause some problems asymptotically. And that's what sort of TMLE or other sort of semi-parametric and non-parametric efficiency frameworks are designed to control, is to ensure that this term is asymptotically negligible. Okay, so out of Hilbert space land for a little bit, back to science. I think we'll maybe all be happy to be back to science. So this is just illustrating a couple scenarios where we've used these methods in practice. So this is a malaria vaccine. So the RTS AS01 vaccine is, as of now, the only licensed malaria vaccine for prevention of malaria. It's deployed in Africa. And here was an analysis we did. This vaccine was developed based on a malaria parasite that's called 3D7. It's a West African parasite. And so it's natural to ask, will this vaccine work as well in West Africa as it will in East Africa, for example, where in West Africa we have lots of 3D7, in East Africa not so much. So we did this analysis that showed basically the vaccine is efficacious against both forms, 3D7 and non-3D7, but markedly more effective against 3D7, indicating this sort of Sieve effect that I've alluded to. So you can also, as I mentioned, study sort of not just 3D7 versus not 3D7, binary J. We can also have ordered J. So this is an analysis where we looked at how far a particular T cell epitope had diverged from 3D7. So it's basically counting the red dots, right? Counting how far we are away from the original strain and studying how efficacy falls off as a function of that. And so to sort of develop a statistical test for this, we proposed basically a linear working model and the log relative risk scale where we project that function onto a linear function and we test the slope of that linear model. So that's what's shown on the right, is showing basically perfectly matched to 3D7 strains here had the highest efficacy. And as you add more and more red dots to that TH3R region, the efficacy falls off, and it falls off almost log linearly. And that's what this is showing here. So the method described in this paper is basically how do we project these black points onto a straight line and then perform inference about that projection. So another bit of work we've done related to the malaria vaccine allows sort of more general projections onto working models. And the idea of this is that Sieve effects almost feel like an interaction. They feel like an interaction between the vaccine and the strain of pathogen, right? It's kind of like this idea that is the vaccine more effective for men versus women or old versus young people. So if we were in a regression framework, we kind of want to evaluate that using kind of cross product terms in a regression model, right? A vaccine by sex indicator and test whether that coefficient is significant. But Sieve analysis is a little more complicated, right? Because the strain of infection is not a baseline characteristic. It's something that happens after vaccination. So you need to be a little bit more careful about how you set up this regression problem. So in this paper, we provide sort of this regression style interface where you can pose a working marginal structural model. So a model that involves maybe your vaccine status, the strain of infection, and maybe other baseline characteristics. And you can pose like a very familiar regression model for that. So if you want to test for a Sieve effect, for example, you would have a vaccine by strain interaction, and then you would be doing a test about beta-3 here to understand whether or not there's a Sieve effect. We can also, extend this to three-way interaction. So does the strength of the Sieve effect depend on, let's say, a child's age? So that's what this analysis was showing, basically that there was no evidence that a child's age impacted the magnitude of the Sieve effect that we observed in the previous slides.
Can I ask, what was M? What is M? Is that a, it's not a time to something here, right?
This little M?
Oh, yeah.
This is, it's a working model. So we're posing basically a working model for your risk of infection or disease as a function of vaccine and, the strain status.
Okay.
Is that Poisson working model?
Sorry?
Is that Poisson working model?
It's a Poisson working model in this case, because we like, relative risk inference. But, we have general sort of Kolbeck-Leibler projections onto your favorite parametric working model that we talk. Oh gosh, I'm already talking way too much. So let's say that we have some ongoing work specifically related to COVID and missing strains and how that impacts this whole story. I gave a talk on it at JSM. So briefly, let's talk a little bit about the current work I've been doing on vaccine correlates of protection. So remember, this is sort of the other side of the Sieve coin, right? Is asking what are these immune responses that are causally related to the outcome? Is it antibodies? Is it T cells? Is it both? So a lot of the work that I've done in this space has been in the context of this COVID-19 prevention network. So basically, I worked in this HIV vaccine trials network that when COVID happened as part of Operation Warp Speed, they pulled together a bunch of existing clinical trial networks to support the phase 3 studies of COVID vaccines. So we worked with 5 companies, Moderna, basically everyone but Pfizer, Moderna, AstraZeneca, Novavax, Sanofi, and Johnson & Johnson. So we worked with these 5 companies. They got funding from the US government. And as a result, we got to insert our own statistical team into their company and ask for a little bit more data, things like collecting sequence information, things like standardizing the way that we measure immune responses, things like standardizing the design across these different trials, as well as just general consulting on efficacy and safety monitoring for those vaccines. So very early on, we published a publicly available statistical analysis plan for how we study this correlates question. And we've sort of been updating that as we've gone along. And in the name of just sort of being very open, if you remember in 2020, there was a lot of skepticism around the vaccine development process. Are we going to approve vaccines that we don't know are safe and effective? What are the study designs that are being used? What justifies them? People like Eric Topol were saying very angry things on Twitter and asking for things to be publicly available. So we tried to honor that. So all our statistical analysis plan was developed in the open. All our code is available on GitHub, et cetera. So one of the key components that is part of this study design for this sort of harmonized design for correlate studies of COVID vaccines is implementation of case cohort sampling. And so this is simultaneously a very sort of useful trick for speeding up getting correlates results and also presents lots of interesting statistical questions. So the idea of a case cohort design is we do these COVID vaccine trials, they enroll 30,000 people. We don't want the labs to have to measure 30,000 people's worth of immune responses because that's very expensive and very time consuming. And also it's pretty much statistically unnecessary when we can use designs like this. So remember in a trial like this, the outcome you can think of as binary. It could be time confined, but it's basically binary. In other words, saying that the power to detect things like correlates of protection is driven by the number of events that you have, not the overall sample size. So if you have a sample size of 30,000, but only 100 observed events, like that 100 is driving your power more than the 30,000. So what we do in this design is we say, okay, we're gonna basically grab all of the cases that we observe in a trial. Those 100 cases, we're gonna make sure that we measure their immune responses because those are the most interesting people. We wanna know, did these people get infected because they had sort of bad immune responses, if you like. And we take all of those cases and we augment those with sort of a random subcohort selected at random from the population. And that allows us to do some pretty interesting things. Number one, we select the random subcohort right away. So we can start shipping samples to the labs straight off the bat. We can start running the immune assays right away. And then as cases accrue, we can send those samples on as well. So it's an expedient design, but one that introduces some pretty interesting statistical complications. The second thing is that you say, well, we're in the context of a randomized trial, so maybe causal inference is quite easy in this study. You're randomizing vaccine assignment, that's great. But the types of questions we're asking with correlates of protection really are more like observational data, because correlates of protection are asking about causal effects of immune responses. And we don't get to control the immune responses that we have. We just give you a vaccine and your immune response is what it is. So when we're answering these questions, we definitely have to worry about things like confounding of your immune response and risk of disease. So we really do need like true causal inference approaches to appropriately address these objectives. And there's lots of frameworks that have been proposed to do this, things like principal stratification. There's non-causal approaches to this as well. The one that I've been working most closely on is causal mediation of this. And just maybe in the name of time, I'll just flash this up here very briefly. What we do in a mediation analysis is we try to do a decomposition of the total effect of the vaccine. So what I'm showing here is like this, the total effect characterized by, again, now ignoring strain for the time being, maybe efficacy over all strains, and we decompose that into an indirect effect. So that's the part of the effect that's going through the immune pathway that we're interested in. So it's saying the vaccine has an impact on antibody level, and antibody level has an impact on risk of COVID, and we're trying to characterize the strength of that pathway. And then all other pathways are characterized by this direct effect that's shown here. That's saying basically what is the strength of all other ways that the vaccine provides protection. And if we can understand sort of our antibodies, relating to a huge fraction of the protective efficacy of these vaccines that we've seen, or not so much, that can give regulators some information about whether this is potentially a good surrogate endpoint. So I have a tech report here and an R package available on GitHub that proposes basically efficient estimators of these that account for this kind of two-phase sampling design. This was applied in the Moderna data, such as the nature of publishing stat papers, that you can have your method published in Science before it can be published in a stat journal. So this was the study of the Moderna vaccine, where we found that the neutralizing antibody marker mediated about 2/3 of the efficacy of Moderna's COVID vaccine. And this was a particularly nice analysis because flu vaccines, which I mentioned, already have this accelerated pathway. Flu vaccines are also estimated to have about 2/3 of their efficacy mediated through HAI titer, the surrogate endpoint that's used in flu studies. So this is sort of saying that, at least for Moderna's vaccine, we think that the strength of correlate signal here is about the same as it is for flu, where we already have these accelerated pathways open. And then briefly, another way of characterizing sort of correlates of protection, a different causal framework, is based on stochastic interventions. The idea here is that mediation is really lovely, but a bit controversial for a number of reasons, one of which is one of the assumptions that you need to identify mediation effects basically requires there to be vaccine failures, or in other words, individuals that you give the vaccine to and they generate no antibody response, so that they basically look like they got the placebo, right? And that's kind of key. Those are the really key individuals that we need to learn about these mediation effects. But for certain vaccines, like Moderna, that's not always plausible. But Moderna, for example, and Pfizer are highly immunogenic vaccines, such that we analyzed a data set of like 1,000 people, maybe 2 vaccine recipients had undetectable neutralizing antibodies. So it's just not, it's a no-go basically to study these correlates effects with mediation frameworks. So in this case, we can use something like a stochastic intervention, and I'll just say a little bit about why I like this estimand. So what is this estimand? It says, what would your risk of COVID be if I give you a vaccine, right? And then I take your natural immune response, the level that you naturally have when I give you a vaccine. I take those antibody level and I add just a little bit to it. So I add delta units of immune response. And why do I like this? Because it kind of, you remember the first regression class that you take, and the way that we're not supposed to interpret regression models, right, is saying that if I increase X by 1 unit, Y increases by this amount of units. And we like wag our finger at you and say that's not the right way to interpret regression models. And indeed, in an associational context, that's true. So this is the sort of formalization of that estimand in a causal in a causal way. So we say, if I take your existing immune response and I just add a little bit to it, how does that impact your risk of disease? And so we end up with pictures that look like this, where we play this across a number of grids. We can project them onto working models if we'd like, and sort of summarize trends across these. And we can identify these types of effects without the sort of strong assumptions that are needed for mediation effects. So this is just a bit of estimation theory that justifies working in 2-phase samples. I think in the name of time, I'll sort of skip over that. So I'll finish just talking a little bit about some computational challenges. And I put this in here because, you know, this represents another area of my work that I'm interested in, which is statistical computing and statistical computing for reproducible research. So when we did this analysis with Moderna, it was quite challenging because basically all of these companies had specific agreements with the US government about what they had to share with the government versus what they could keep to themselves. And Moderna, being the new company that they are with basically only 1 vaccine product, was very cagey about sharing their data. And so they didn't want to share any of their data with us until they had a full BLA for their vaccines, basically until their vaccine was fully approved. And yet we had to figure out a way to run all of our correlates code on their data without ever touching that data ourselves. So you can think about this like, you know, nightmare scenario where you're working with a statistician at Moderna who only knows SAS, and you're handing them like 50,000 lines of R code and saying, please, can you run this for us so we can get results? So that's sort of the challenge we were up against here. So we worked, we had a great team. There's about 12 programmers and statisticians that worked very hard to have sort of a synchronized pipeline. So we worked with Moderna to build a synchronized environment using AWS. We have sort of fully reproducible reporting using things like our Markdown and the Bookdown package. We use ROM for our package management, so syncing up package versions across different systems. We had a continuous integration where basically we did a ton of sort of practice builds of this report using a continuous integration service. We had formal double programming of these results, so including like very complex machine learning analyses. So that was an adventure. And in the end, after much patience from this lovely Moderna statistician that we worked with, we were able to generate this report that ran to over 1,000 pages without ever touching the data ourselves. So it's a pretty cool sort of illustration of the power of the types of tools that we have to do reproducible statistical computing these days. And then, the nice cherry on top was that, when we published our preprint, we got to hear Tony Fauci talk about it as part of a White House briefing, which was pretty neat. Not everyday occurrence for us statisticians. So this is ongoing work. We are working basically ticking off all the different companies. So we've already done this for the Johnson & Johnson vaccine and Novavax. Those preprints are available. The AstraZeneca one will probably be available this week. And then we're going back to hit on different questions. So like one project I'm leading is basically saying, do we need to measure one particular antibody response, or is there benefit in adding more assays, different immune assays, to come up with a stronger correlate? And then we also have ongoing collaborations with Sanofi and Sinovac as well. So where are we with COVID-19? So basically, this was a very stressful analysis for a number of reasons, because the community sort of assumed that antibodies were what was going to be important. And then we had to do this analysis, and like my greatest fear was that our analysis was going to show that antibodies weren't important and like everyone was going to attack us. Thankfully, it didn't show that, so that's great. And neutralizing antibodies have basically continued to be sort of a pillar stone for regulatory policy making. So you've seen FDA sort of quietly expand indications for vaccines into pediatric populations. That's mostly based on neutralizing antibody studies. When we're now talking about approval of variant-specific boosts, that's mostly based on neutralizing antibody studies. So this is a figure here showing basically that justified was a key piece of evidence for justifying the need for a third dose, showing that basically against Omicron, based on 2 doses, we had essentially no neutralizing response to Omicron with a third dose that boosted entirely. So, so antibody continues to be pretty important in the COVID story. So there's lots of open areas for statistical methods development in this. So mediation analysis has lots of like fraught assumptions that I didn't describe. So sensitivity analysis, causal sensitivity analysis, I think is key there. Now we need to get into the world of longitudinal studies, right? Not just of kind of your peak antibody level, but how that decays over time. So finding the right formulation of causal frameworks to ask and answer those questions, I think is pretty interesting. I'm also interested in, prevention studies, not through vaccination, but through monoclonal antibodies. So I have some ongoing work in that area. And then basically there's a huge need to develop software to get these tools, to the other research groups who are performing these types of correlates analyses as well. So I think I'll stop there. Again, please feel free to follow up if you have any questions. And thanks so much for having me. Looking forward to meeting the rest of you while I'm here.
Okay, thank you very much, David. We have some time for questions. Yes. Go ahead.
Yeah, I got a question about, so you have this model on the mediation analysis slide where you kind of look at vaccine efficacy partly as a function of how much we've moved from your natural immune response. And I guess that connects to neutralizing antibodies predicting quite well how vaccine efficacy will perform. But just, I think, so for people who say might have much lower natural immune responses, like people who, for example, are on immunosuppressants or something like that, that shift might be just as large, even if their neutralizing antibody level is much lower. So for those groups of people, you might have a much less predictive neutralizing antibody response. Is there a way to measure people's natural immune response and then the relative change that would allow us to like look at those groups of people?
Yeah, no, it's a great question. I mean, so there are still assumptions here. Like you have to assume that, like, let's say for an individual who is immunosuppressed, you have to assume that it's plausible to shift their immune response at all. And like, maybe it's not, right, with the vaccines that we have. So there are still, that sort of assumption is underlying that, but that's sort of a weaker assumption in some sense than what mediation requires. So to answer the second part of your question, which was, do we measure? So what we measure is somebody's natural immune response, right? But the shift is hypothetical, right? And we need that hypothetical shift to be realistic for any of this to make sense. So I agree, there's going to be specific populations where even this type of estimate isn't quite going to get you what you want.
Else?
So can I follow up just a little bit, sort of the same theme. So if you try to get into like precision vaccination here, so you got another covariate, and you sort of know it might not, vaccines might not work for one group quite as well as the other, and this, but the causal framework is always focused on marginal effects.
Yeah.
So how do you sort of square this sort of causal equals marginal with sort of desire to do different things for different people?
Yeah, great question. I mean, so in these analyses, we do, every one of these analyses we also do stratified by key characteristics that we care about. Old versus young, do we have the same strength of signal, sex, race, ethnicity, but precision is maybe a little bit different of a question. I mean, because maybe there you could get into, can you have adaptive vaccination policies that say, if your titers fall below this level, you need a boost kind of thing. And I think that's ideally where we would like to be. But as far as I know, we don't have great data for that because we really capture these markers at like one time point after vaccination. And then we had planned to measure them longitudinally, but like EUA vaccines kind of blew all of that up. So right now we're kind of working with US government and these companies to understand like, what can we salvage from that? How much information do we have about these mixed dose vaccines and when they got vaccinated and so forth? And those are the type of questions we might want to try to ask there. Yeah.
I have two quick questions. The first question is, when you were presenting on the second slide, I couldn't read from here. Was it a picture across delta on the next slide? Was that the?
Yeah, exactly. Sorry.
So the way you would normally present these kinds of results is over choices of delta, and that delta is a range that you find plausible. Is that the way?
Yeah. Yep.
And then just on this, now that's three questions because I'm sorry. But you're doing it on an additive scale, but I could also do it on maybe on a multiplicative scale instead, M1 times e to the like some type of, like I'm increasing the M1 by some percentage as opposed to an additive. Okay.
Yeah. So I mean, you can do there are sort of smoothness constraints on what you're allowed to do here, but yeah, you can think about any kind of shift like that. Gotcha.
And then on the two-phase part, I just had a quick question. So you had a statement about random subcohort, and I was just curious. In practical setting, right? You want to have a random subcohort, but you're going to have non-response, and you're going to have. And I'm just curious, like in this setting, how much, when you chose that random subcohort, how much were you dealing with non-response rates and missingness, et cetera, when you did that?
Yeah, thankfully, like people don't get much choice because we do blood draws on everybody post-vaccine. So this is happening. The blood draws happen basically the day you receive your second dose. Before you get it, we take a blood draw, and then 2 weeks after the second dose, you come back in. We take a blood draw and we store those samples, and this is just deciding which samples we pull from the freezer to ship to the labs. That said, working with Sinovac on their study, which happened in Brazil with the Instituto Butantan, it was a particularly chaotic time in Brazil during early COVID. And so there were problems with missing samples and mislabeled samples and a lot of, the practical issues that come up with, true responder bias, et cetera, so that we had to work through as well. But for the most part of the US government trials, this was pretty well controlled. There would maybe be like one sample missing out of 1,000 or something, not enough to really worry you.
Thanks for a great talk. I have a question. Basically, so you said did not analyze data from Pfizer and BioNTech, right?
Yeah.
And they use Bayesian beta binomial model.
Yeah.
Any kind of experience to share regarding the
Regarding
choice
what?
of methods?
Which controversy do you want me to comment on? I don't know. I'm not. I thought Pfizer's design was fine. I thought the Bayesian analysis was fine. They have a funny little, nice teaching moment mistake about their prior that they use that they can point out to anyone who wants to use it in their Bayesian class. But I think like one of the first things I did, because Pfizer was the first one, and they were like the one that didn't contract the USG, and so like people were very skeptical of that. And so and they had 5 interim analyses planned, and people were like, that's a lot of interim analyses, basically. And So what I did was basically, you can look at the case splits, case conditional, and see when they would have rejected, and it lines up basically with frequentist bounds. Like I think it was like O'Brien-Fleming basically in a frequentist setting, modulo like a fraction of a degree of power here or there. So I don't know. I don't see that as a huge controversy. I think it's a little frustrating that Pfizer chose to strike out on their own, and it was a strategic decision on their part. It would have been better, I think, for science had they engaged with this process, because basically they're benefiting from all the analyses we did and the U.S. government paid for, but they didn't want to play ball with us. So sorry to any Pfizer shareholders. Yeah.
Yeah, I have a question about this patient recruitment. Is this trial conducted only in the US population, or is worldwide involving multiple countries? And how to deal with the heterogeneity across different populations?
Yeah, so it depends on the company. So I think Moderna was only US sites. AstraZeneca was mostly US, but a couple South American sites. So it just varies. And thankfully, well, I don't know, thankfully, but the fact is that the phase 3 trials were so early in the pandemic that sort of that SARS-CoV-2 hadn't diverged much from Wuhan yet. But for the later trials, that becomes a really complicating factor in interpreting the results, is that you have a Wuhan-like vaccine that's being deployed in like the US, where Wuhan is prevalent, and it's also being deployed in Brazil, where Mu is prevalent or something. And so basically, so Ensemble, the Johnson & Johnson study was like this actually. So basically we have to do all the analyses that we do it sort of pooling over regions. And we don't love that because it's harder to interpret. And then we do country specific analyses as well. So for that study, it was really South Africa was the outlier because they had a beta variant emerge, and that was basically a precursor to Omicron, a lot of the same mutations that were problematic. So we kind of had to study that separately from the other countries as well.
Yeah, so as part of design, when you do this randomization, you do the randomization across all the individuals from different countries where you have country-specific randomization. So when you do this country-specific analysis, if there's no, I'm just thinking about which level of the randomization has been conducted. Like if you do a country-specific analysis where, on our hand, the randomization is overall plan rather than country-specific. How do you justify the confounding for the specific country, right? So because you don't have a complete, perfect randomization in a given country.
I mean, I think I'd have to go back to the protocols, but I'm pretty sure that these will use study site specific or study site stratified randomization. So I think that's accounted for. I would need to go back to the protocol, but that's how at least in HIV studies we typically do.
I got another one.
Sure.
So for that malaria study, I think, so as I understand it, you kind of have two strains commingling at the same time, right? Like they're kind of the Western and the Eastern one are sort of constant over time. But obviously it's very different from the COVID situation where suddenly one just completely kills the other, and that's within a relatively short calendar time window. So does the COVID scenario present problems relative to this?
Definitely, yeah. I mean, you have a perfect, in some situations, you have perfect confounding of sort of time since vaccination and emergence of a strain. And so you need sort of a lot of variability in when people got vaccinated across different regions, so that, what you can see is that, the vaccine efficacy is worse against Omicron, not just that efficacy overall wanes, right? So like if Omicron emerges 8 months after people get vaccinated, is that waning efficacy due to the fact that they got vaccinated a long time ago, or is it due to the new variant? So definitely it's harder when you have that sort of.
And in relation to that, I know currently you're just baseline covariates, right?
Yeah.
So in relation to that, is there ideas to extend that to time varying covariates, time from vaccination?
Well, yes, I think so. I think, with time varying covariates, you have to ask what problem are we trying to solve? There could be a number of different problems. So for example, in this, wherever it is, this extension that I didn't get to talk much about, Here we have sort of missing strain information. Basically, they try to run sequencing of the blood samples after people are infected, and sometimes that sequencing fails. Sometimes that sequencing fails due to low viral load, and we're a little worried about that because maybe some types of new strains emerge that have naturally lower viral load. So like it becomes a sort of harder, missing data problem where you have these sort of time varying covariates, like how much viral load is associated with that, what is the symptom profile associated with that. And you need to use that kind of information to control for this missingness. And then certainly, like as I'm sure you've seen in studies of observational, studies like that CDC is putting out, for example, there they are looking at things like time since vaccination because they have sort of more observational cohort data. Here in a randomized trial, these trials recruited like super fast, so we don't have a lot of variation in that. Because recruitment only took four weeks to get 30,000 people. So you just don't have the variation to be able to do much about that.
Let's quick follow up on that. Sure. So, does that affect when you're starting to bring in time varying covariates, does that affect the causal interpretation of some of your modeling and so forth?
Not the interpretation. Like I think we're still going after the same estimates, but it impacts the identification. So identification becomes more complicated because we have to sort of disentangle multiple levels of confounding.
Yeah, developing like so you end up with a time varying covariate that now has all these potential outcome types of.
Yeah, right.
Any other questions? Sure.
So your cause specific hazard framework, right? So when I think of cause specific hazards, dying of cause 1, cause 2, cause 3, cause 4, and they're all potentially going to happen.
Yeah.
But here, the only variant floating around is Delta.
Yeah.
And so there's no possible getting infected due to Omicron. So there's no sort of diminution of the actual incidence of the Delta thing because of the existence of Omicron. So does that you handle that okay? Doesn't seem quite, so it seems slightly different, right?
It is slightly different. I think both in terms of the way we think about modeling, it's not like stroke versus heart failure where you have very different etiologies, right? It's basically the same etiology where you engage in risky things where you might get infected with SARS-CoV-2 and sort of the cause specific element of it is just like where are you located in time and space and what pathogens are circulating around you there. So I think, thinking about how to model these things is quite different than kind of more classical, maybe cardiovascular related competing risks. In terms of, so you're then concerned about sort of scenarios where there's not
There's nothing competing.
Nothing is competing in a particular region or something.
Sort of focusing on cumulative incidence will be different in one region where there is in another region where they are competing.
Right. Yeah, I think that's definitely needs to be carefully considered, and it comes back to previous question about different countries at different times, and is it appropriate to pool over them over time? And so, you can end up with this phenomenon where beta emerged in South Africa, and over the course of the study, basically you have perfect confounding of beta's emergence and people who live in South Africa. They have different gut microbiomes, for example. They have different physical activity profiles. So you have this sort of perfect confounding of the people who are receiving the vaccine and the types of pathogens that they're exposed to, and that could be potentially problematic, right? If the types of people who live in South Africa are meaningfully different in terms of how they react to vaccines or the risk of COVID from the US. So there would be a situation where pooling over regions would be quite potentially problematic.
Interesting.
Yeah, thanks.
I know we're over, so feel free to run if you need to, but any other questions?